As Janos mentioned, I'm Mark Somogyi, and I'm the product manager for the biologics software at Chemexus. In the first section of this webinar, I'm going to give you an overview about biomolecular structural data management powered by Chemexus tools. First of all, I would like to outline the agenda for you. I will start by an introduction about Chemexus, and later I will transit into discussing the opportunities and challenges of biotherapies. Here, I would like to clarify to you what is considered to be a biomolecule, what people who are inventing such novel bioentities are thinking when they are working with biomolecules. And later on, I will give you an overview about the features as well for handling complex biopolymers. As I promised, I will start by outlining some facts about our company. So Chemexus is a leading cheminformatics and bioinformatics company founded in 1998, of which headquarters in Budapest. And we also have a couple of other offices worldwide in the US and also in Switzerland and the Czech Republic. Our company is 100% privately owned, and it now counts more than 250 employees worldwide. Our versatile product portfolio consisted of more than 30 software products, which fit to the different stages of the drug discovery workflow, starting from target validation to lead discovery and design till clinical trials. For each of these drug discovery stages, you can find the appropriate software package that can help your work. Our product portfolio consisted of tools for designing, searching, registering, and analyzing drug modalities, help scientists to manage chemical and biological data. And now I would like to take you to the biotherapeutics world and really take a look at the drug development from a different perspective. In the past decade, we have seen that chemistry has started to expand rapidly into the realm of biologics, resulting in novel bioentities, for instance, synthetic peptides, therapeutic RNAs, and their conjugates. Companies have started to shift their pipelines towards these novel biomodalities because they are offering promising therapeutic approaches to treat unmet medical needs. And besides this, companies aim to keep their original pipelines consisted of traditional therapeutic approaches, which includes recombinant protein and antibody-based therapies. Some of you might ask the question why companies and the market choosing biotherapeutics and started to shift their attention towards this direction. And I would start with a pretty simple answer. It's simply because that novel biotherapeutics offers good alternative strategies to modulate biological targets. And since chemistry started to expand rapidly into this space, scientists inventing novel chemistry in order to improve biological chemical stability and also specificity and potency at the same time. Besides this, there is a continuously increasing chemical space, which allows us to introduce novel ways to deliver biomolecules to the target organs. As a result of this, companies need to combine special expertise within and all across the organizations. This is where we can say that chemistry will meet biology, and also certain specialties and expertise needs to be outsourced. And this is where third parties are stepping into the picture and collaborating with the organizations. So as a result of these trends and circumstances, there is a tremendous amount of data management challenges ahead for the already established informatics systems. For instance, one of these challenges is to understand the biological and chemical structural context to a sufficient extent. This is actually the #1 issue for those organizations when you need to combine chemistry with biological knowledge within and all across the R&D departments. And same is true for the case when you outsource a portion of work in the form of contract work or contract manufacturing, when you assign a third party to create a custom synthesis work. And the other issue which is recurring is the Excel spreadsheets, which creating data silos, which actually make the whole R&D process less efficient and productive. And furthermore, due to the continuously increasing chemistry space, there is an increasing complexity of these biomolecules. Different permutations will actually result in decreased visual interpretation efficacy, and also scientists need to spend more time on designing structures, basically. So I have mentioned the term biomolecule a couple of times, and it is time to specify what the people think when they say biomolecule. And you can see on the screen a cyclosporine A peptide, which has the chemical and the sequence-based representation next to each other. The cyclosporine A composed of multiple artificial building blocks, of which has chemical modifications on the backbone and also on the side chains. The point here that these information pieces alone are not sufficient to perform a productive work. So to perform a thorough analysis on these structures, you need to have this information together somehow in one. Because if you are performing the analysis, first of all, you will do sequence comparisons, and later on you will drill down into the chemistry. And the sequence-based representation might not be verbose enough for those who are not using the given nomenclature. Maybe it's not trivial for them to be used. The other example here is from the public literature. This is a special one. It is called the peptide oligoconjugate, of which has two compartments. The RNA compartment is highlighted in purple, and the other one is highlighted, the peptide, which is responsible for penetration, cell penetration, is outlined in green. And here we also encounter a similar scenario, but on a different modality, because if you want to perform a thorough analysis, first of all, you will do sequence comparisons and later on drill down into the chemistry details. So this is also a scenario when you need a combination of these two words to perform an efficient work during the R&D process. To address and answer to all of these challenges, we offer a set of tools that can model and register any biological entity, ranging from chemically modified entities, the ones that we have discussed, peptides, oligonucleotides, and their conjugates. We also support biological entities which has purely biological nature, for instance, proteins and antibodies. And scientists can also capture tools and regions which are necessary to be tracked in order to understand the behavior of the end product. That can be either a protein or any biological nature entity. I mentioned that organizations need to understand the chemical and the biological context at the same time. And this is where we see a huge variety in terms of how companies describing their biomolecules and the complex chemistry around this. And to standardize these complex biomolecule representations, we use HELM as one of the early adopters of HELM standard. We can use our software to use HELM for deciding upon the uniqueness as well. HELM is basically follows a combined approach. You will have the sequence-based interpretation and implicitly the chemical meaning behind this monomeric makeup. So indirectly, you can set the nomenclature and also the chemical definitions behind these building blocks. Besides this, I mentioned that chemical structures are also necessary to a certain extent. And it can happen that within the organization, scientists are creating these chemical structures in a slightly different way. And to achieve uniformity within the data management systems, we allow customers to use our tools to quickly modify in an automatized fashion the chemistry around these biomolecular structures. We also have tools for visualizing and designing entities. The image that you can see here on the slide shows Bioready, our biomolecule structure editor, which is dedicated for depicting and modifying chemically modified entities, such as peptides, oligonucleotides, and their conjugates. On the top left, you can see a simplified representation of the cyclic peptide, which has a chemical modification on the C terminus. And in parallel, you can review the chemical structure, which is generated based on the simplified representation. And using this combination of this use, the scientists can easily interpret and share biomolecular structural data with their colleagues and within the organization. On the right-hand side, you can see the library panel from where you can utilize chemical building blocks to introduce novel chemistry and unique sequence patterns to your biomolecular blueprint. I mentioned that there is a monomer dictionary which resides in a monomer database, and this is an interface called the Monomer Library Manager, from where you can maintain and track the chemistry behind these monomeric makeups. And once this monomer database is set up, it's an easy way for scientists to manipulate the structure and also to obtain the full atomic representation if it is necessary from the HELM strings or from the sequence-based representations. If you want to manipulate and customize your monomer dictionary, then you can use the embedded Marvin.js to draw the atomic level and the chemical definition for these building blocks. If it is also an option to use generic monomer database based on the recommendations of the HELM standard. This is all that I wanted to share with you. Thank you for your attention, and I will be happy to take any questions in the Q&A.
Yeah, so thank you, Mark, for the first part from the Chemexus side, and on our side from Discngine, I will talk a little bit about the reasons why we started to partner up with you, and especially in the context of one of our products, which is called Free Decision. So I'll go a little bit into the details there, but in general, the main challenge that we're trying to address is to properly handle the complexity of biomolecular structures, so not just small chemical structures, but biomolecular structures and the 3D structure information contained within them as well. On top of that, but first, a few introductory slides to Discngine, in case you haven't heard about it. We are a Paris-based company, have been around for quite some time now, and are steadily growing, even though we are not as big as Chemexus now. And we are active worldwide, working for a few customers, mainly in the pharma and agrochemical industry. And our focus is a little bit more towards very early stage drug discovery, so molecular discovery on different scales and different areas there. The one I will focus on more today is more the chemoinformatics and bioinformatics scope. But just to be more complete, we also have teams working more on the real science, so the data acquisition, sample management, etc. in the lab. And we also have lots of things around visualization and data analysis in the scope of early stage drug discovery projects. One important thing is that Discngine is not developing molecules, so we're not a pharma company, but we're helping our customers, which are just the pharma industry, to develop either their own tools, services, or provide products to answer specific questions. And one of the particular things about Discngine is that since the beginning, we've been partnering up with lots of historical and new partners. So Chemexus is one of these partner lists that joined us lately. But we have quite a long track record of working together with people from Biovia, so former Accelerus, or Cytigic, Oracle, TIBCO, on the Spotify integrations, and many more, as you can see here. And I think that's one of the guiding themes at Discngine, that we're trying to not to invent everything, but to try to integrate the best of breed technology and where we see its needs. And that's also why we started the collaboration with Chemexus here. So without further ado, a few facts about Free Decision, which is our SaaS platform for 3D biomolecular structures. The main aim here is to gather and digest the complexity of data that you have within 3D structural data, So when I say 3D structures, it's not just chemical structures, it's really a full protein structure, even with a ligand or without a ligand in there, and trying to understand what is the protein behind the scenes, where are binding sites located, how does a ligand bind there, etc. The main aim here is to not just to propose yet another PDB-like service to customers, but to allow customers to register their own experimental structures, but also in silico structures, models, in order to use them at a later stage in downstream projects. In terms of information content, or the main, some of the main purposes of free decision are listed here. So first of all, we want a centralized knowledge hub to really have everything structured well together, so then we can do quite powerful queries and searches on them. And by feeding in new information, so new experimental structures being resolved or new models being created, the system actually automatically has more information content, so it learns without using the AI buzzword. It learns basically from what you put in there for new projects. One paramount thing is as well is we try to improve the collaboration between interdisciplinary teams. So this has been mentioned a bit by Mark, where we, on our side, we have chemists, we have crystallographers, we have molecular modelers, and we also have lots of biologists interested in the tools. And making all of these people communicate together is quite challenging, and it usually involves heavy emailing with lots of files, etc. So trying to improve the way people work in there. And on top of that, typically try to ease the access to some of the complex knowledge that is contained within the data that we handle here. To give you a rough idea of what is in free decision, because I kind of do a demo during the webinar here, is that we gather all the structures from the public domain, so from the RCSB PDB, but also for particular private customer private structures, so they would be basically imported in the beginning, and we can also handle any in silico model that you would like to put in there, so homology models, refined docking models, protein designs, etc. They can be registered into free decision, and on top of that, we associate each structure and each component of a structure to an actual sequence. If it can be matched to an actual canonical UniProt sequence, then this is done. And on top of that, we add lots of annotations on the structures and on the sequences. And once this has happened, we analyze the structure more thoroughly, identifying what are the ligands binding there. So that's small molecule realm, how they interact with the protein. So in terms of types of interactions, vectors of interactions, etc. We identify putative and known binding sites, etc. But we also integrate, for instance, chemical data and assay data, etc. Plus lots of additional things that you could add then in there. So those are just 4 screenshots. Again, I cannot show it, but the main message here is that there are several use cases that you can address by using free decision. So first of all, you can very easily structure, when you have lots of structures available and you're lucky to have them, you can structure this data together into projects, easily overlay things, expose things to chemists so that you get new design ideas. You can even run some on-the-fly docking, get some estimation on how well your ligand fits according to contact statistics that we have within the structural database behind the scenes. And you can also go towards a more sequence-based analysis, like whether a model organism might actually suit your experimental tests, or whether there's some mutations within the binding site of interest. The main message here being is that right now, historically, free decision has been developed with the small molecule design concepts in mind, even though very early on we had some POCs in order to enable people to handle peptides as well. So we have workarounds, so people started to assess this, and we have lots of customers asking for these types of functionality. And we also had lots of requests regarding antibody sequence and structure handling. So the same here, we had lots of POCs in the past to enable some of these functions, and some of them are integrated now. But then we decided to look for what would be the best option to get a production integration of these functionalities, and that's why basically we started to communicate with Chemexus, and especially the biomolecule registration toolkit that Mark talked a little bit about before. So on the roadmap for this year, so this is not released yet. So what you're hearing here is what we will work on this year. Is that we're trying to enable the same as you do with small molecules in free decision right now. So meaning superpositioning, correct capturing, registration, and visualization, but also editing of peptides. We want to enable a full peptide support within free decision, so that a peptide is really recognized as a peptide, and that you can actually act chemically upon the peptide and see what changes would be caused by your modifications. The good thing about this is there's a minimal change only necessary in the UI, so it's mainly integrating things in the backend. The second work track is linked to for antibody registration. So we've done, as I said, a POC before on our premises, but we would like to push that further using the toolkits from Chemexus. And here we envision, given the feedback we had from the first integrations that we've done, we will go for dedicated analysis workflows and dedicated UIs in order to allow people to, first of all, register antibodies, but also do typical sequence searches, particular motif searches on CDR regions or regions of interest, associate the corresponding structures to them, etc., and also enable some basic superpositioning in the beginning. So yeah, again, as I mentioned, for the peptide support, we will try to target these ones here. So the most important ones being the correct registration depiction, and the downstream would be then the search. And in addition, would be the cherry on the cake, where we could use, for instance, tools like Bioready, enable people to modify peptides in the binding site themselves. On the antibody registration side, like I mentioned before, a little bit the same. Again, we already have some functionality on the sequence search, but here this time around it would have to be changed towards the biomolecular toolkit. But on the longer run, what we are aiming here for is really to enable more structure to property handling functionality that you would like to have with when you're optimizing your sequences, or when you're optimizing ADCs, for instance, in here. On the integration, so that's more technical aspect, is that right now we are undergoing a heavy backend revamp on the free decision side, which will make the integration much easier than before, let's say. So the way free decision works is that you have a user here that interacts through web browser, either through Chrome, through the UI, or through a REST API with free decision. And free decision is basically now a set of services exposed through REST API. We use Nest.js for that. That is linked to a quite large database behind the scenes, and a growing number of microservices. Yeah, so we opted for microservice architecture behind the scenes for various reasons, and one of those reasons is that we can easily add in plug in new things. So, for instance, toolkits from Chemexus are quite easily to be added to the scope of the application here. On top of that, we're doing a correct abstraction of those things, etc. And just as an example, so that's outside of the biomolecular toolkit world, etc. But for this new version of free decision that will come out by the end of the month, we already have an integration of the Marvin.js sketcher. Actually, we had it before, but anyway. So that will become the new default sketcher for pre-decision. We also replaced the molecular cartridge for small molecule searches, so that's going to be Coral from Chemexus. Starting with this new release. And we also have an integration already of quite a lot of different services exposed by JChem microservices. So among them is the molecular standardization, but we also have some property calculations in there exposed that are of interest for us to correctly correct ligands that come from non-refined PDB structures, but also assess protein ligand contacts in a more accurate way in here as well. So we already started to integrate some of the services. We know the challenges and the advantages of doing so. So we're quite confident that this will be a quite successful thing for this year. And I'm aware that I couldn't, especially for those who know Chemexus but don't know Discngine and free decision, I couldn't talk a lot about free decision. So don't hesitate to go to this URL, to Discngine, if you want to learn more about the tool itself and what it can do. And I'm happy to take questions, the same as Mark.